Okay, welcome everyone. This is our fourth symposium titled Hormonal Contraceptives and Cognition in the Mind the Gap series of symposium. And today we have three incredible speakers that we can't wait to hear from. Dr. Adrienne Beltz, Dr. Birgit Derntal, and Dr. Marie-France Marin. During the talks, we encourage that you share what you learned using our conference hashtag WHRC2002, and tag us at Research on WH. Before we begin, I want to recognize that we live, work, play, and participate in community on the unceded, ceded and traditional territories of the 203 First Nations, along with 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I am joining you from Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, Tsleil, Yautut First Nations. We want to thank Michael Smith, Health Research BC, CIHR, and Elsevier for sponsoring this conference, for the support and partnership we received from the University of British Columbia and the IGH Sex and Gender Science Training Network. We also want to thank all of the individual donors of the Women's Health Research Cluster who help us work towards sex and gender health equity worldwide. If you aren't familiar with the Women's Health Research Cluster, we are an international multidisciplinary network of women's health stakeholders who work together to ensure women and girls can live equitably healthy lives across their lifespan. We have lots of events outside of this conference, annual trainee awards, working groups, and more. And we welcome you to become a member, join a working group, or get in touch with us in other ways and engage. And so without further ado, I would like to introduce you to our very first speaker, Dr. Adrienne Beltz. Dr. Beltz is an associate professor of psychology at the University of Michigan. She is a quantitative developmentalist who studies sex hormone influences on the brain and gendered behaviors across the lifespan. She specializes in ovarian hormone, links to cognition, and psychopathology, and uses innovative methods to reveal when women are and are not well represented by averages. Dr. Beltz received her PhD in psychology, specializing in social, cognitive, and affective neuroscience from Penn State University. And she completed a postdoctoral position at the same university focused on quantitative methodology before joining the University of Michigan faculty in 2016. Her research is funded by the National Institute of Health, Jacobs Foundation, and James S. McDonnell Foundation. She has also received awards from the Association of Psychological Science, as well as Division 5 Methods and 6 Behavioral Neuroscience of the American Psychological Association. So Dr. Beltz, when you are ready, we are looking forward to hearing your presentation.
Great. Thank you so much for that introduction. It's a pleasure to be here this Can you hear me all right, by the way? Can you hear me? Okay.
Yes, it's perfect. Thank you.
Okay, great. Sorry for not asking first. This whole seminar series has really been so much fun for me to tune into and see the faces behind all the work that I've been reading for so many years. And today, as part of this last symposium, I just want to thank the organizers, especially Jesse and all of the funders for making all of this possible. I also thought that maybe we could have a little fun. And I'd like to start by talking about my own journey, because I feel like for so many of us doing this type of work, it really has been like a personal and a professional journey. So I'd like to share with you, let me see if I can, there we go, how everything started. It started well, right? About 10 years ago for me, this was a 2015 paper, which means data collection and the work was a couple years before. So in 2013, I had the great pleasure of working with my graduate advisor, Sherry Berenbaum, and the great Betty Hampson, who we all heard from in our very first keynote for this series, on a paper on oral contraceptives and cognition. And I'm going to talk a little bit more about this paper in a bit, but this is the paper highlighted in one of those APA Division 6 awards. And I was like, oh, I've totally found my track. This is going to be my thing. But 10 years later, how is it going?
This is a bogus study. 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 Because if
I'm glad you said that.
Because if they showed differences, that would be sexist and stereotypical.
So about 10 years later, critique and public influence on the work that we're all doing here is a bit stronger. And I'll be honest, when this clip first came out, and they are talking about the study that I'm showing below, which came out a couple of years ago, I was a bit mortified, concerned maybe, and now I show this clip in all of my talks. So it's turned out to be a big win. But I'd like to frame the rest of my talk kind of in this notion, kind of how it started, walk you through some of the ways in which I was thinking about hormonal contraceptives and cognition about 10 years ago, and some of the things we're doing now and the ways that we're currently thinking about this. All right, so how did it start? We really started about thinking about averages, thinking about group differences. I actually came or found my way into hormonal contraceptives because I'm interested in gender, right? Sex and gender differences in cognition. So what I'm showing you here is from a larger view, you'll see the effect size in D on the x-axis and a variety of different cognitive domains on the y-axis. And I'll highlight that on average, right, men and women, and I apologize, most of this literature is on cisgender men and women. And so when I talk about the existing literature, I'll often use that language. But in talking about the implications of hormonal contraceptive work going forward, it's important to acknowledge all people who menstruate or who have uteri and use HCs. So I'll use that terminology when thinking about work for the future and future potentials. But in reviewing this literature, men and women don't differ on average in overall cognitive ability, but there are some robust sex or gender differences in certain cognitive domains, right? Certain aspects of cognition. And I've always found this to be very interesting. Where do these things come from? Clearly from multiple different influences. The biggest one is in three-dimensional mental rotations, and this bar is shown in blue because this is a domain where, on average, men tend to outperform women to a pretty high degree. And this is especially three-dimensional rotations, so seeing an object and kind of manipulating it in three-dimensional space in your mind. The gender difference is much smaller for two-dimensional rotations, for instance. And there's also lots of areas, and I'll just draw your attention here to some of the verbal areas and different aspects of memory, right? Many of these domains you could break down even further, where women tend to outperform men on average. And so I'm going to talk a lot about mental rotations today and a little bit more about some of these verbal memory in particular. So I'm really interested, as I said, in menstrual rotations, particularly influences on this gender difference, such as gonadal hormones, and didn't start in hormonal contraceptives about 10 years ago, really interested in puberty. Puberty as a period of second brain organization, right? Cheryl Sisk in her lab and several others, more than 10 years ago now have experimental evidence from Syrian hamsters, and it's been shown in other rodent models as well, that the brain seems to have this declining sensitivity to gonadal hormones throughout development, meaning that around puberty, when we see these large increases in gonadal hormones, animals who go through puberty earlier seem to have a window that's more open, right? So greater sensitivity to those gonadal hormones than maybe animals who go through or have gone through puberty later. This means that whatever behaviors matter or whatever behaviors are linked to those particular hormones, so that would be maybe testosterone or other androgens in male animals, would be impacted by, would be more sex typical in animals who go through puberty earlier than later. And so this was really highlighting a way in which a mechanism through which gonadal hormones might be influencing some of these behaviors like mental rotations that there's some suggestion even in humans are linked to testosterone. So we looked at this in human males and we found what we expected with respect to mental rotations, particularly three-dimensional mental rotation skills, that early maturing boys who went through puberty earlier, had better mental rotation skills than late maturing boys, even into adulthood, right? So this effect seemed to persist. It wasn't a huge effect. It wasn't completely explaining their mental rotation skills, but we were seeing some signal there. We weren't seeing signal, though, for girls, not with mental rotations or not with things like we used some word fluency tasks that we thought might be estrogen linked. We didn't see any effects, but this was kind of our first pass at this. And so we wondered, well, what about hormonal contraceptive use? What about the menstrual cycle? Is this, are these hormone effects kind of co-occurring or overlapping with potential pubertal organizational effects in ways that would make us miss them in this particular puberty study? And so we said, sure, hormonal contraceptive use, it kind of dawned on us, is a natural experiment, like so many other hormonal or different periods in the lifespan across the menstrual cycle, menopause, pregnancy, and then hormonal contraceptive use, menopausal hormone therapy are all ways in which individuals might experience or might exogenously impact their neuroendocrine milieu in ways that we could pick up and ways that we could study. So we said, yes, we'll do this first pass, try to understand these hormonal contraceptive influences on cognition, and then come back and pick up our studies of puberty, right? That was 10 years ago. That was more than 10 years ago. We're still trying to find out what's going on with hormonal contraceptives and cognition today. I know we've talked about this a lot throughout this series, so I'm not going to talk about all the different forms of hormonal contraceptives. I'm just going to highlight two that I'm going to mention a bit here today. The first and the most widely used in the US and in Europe, in North America and in Europe, I should say, is the combined pill, right? So this is a pill that contains a synthetic form of estrogen and a synthetic progestin. These progestins have varying formulations. Typically, they're taken for 21-ish days of an active pill and then about 7 or so days of a placebo pill, during which there's a withdrawal bleed that occurs. And oral contraceptives have their contraceptive action by suppressing ovarian hormone function, and thus it suppresses also endogenous or lowers endogenous hormone levels for things like estrogen and progesterone. But it's hard to compare. Those endogenous levels are hard to compare to the exogenous ones in the pills because they're different hormones in both their type and their potency. And we don't yet have great ways of measuring them, although the field is changing. Contrast that to hormonal IUDs, which are a progestin-only formulation, right? So there's not estrogens in hormonal IUDs. And right, this is a transdermal application into the system, and hormonal IUDs don't have their contraceptive action by suppressing ovarian hormone function, but rather by changing the local morphology where they are to prevent a pregnancy, for instance, by thickening cervical mucus. This is one mechanism. So this means that hormonal IUDs don't necessarily inhibit anything with respect to, or suppress anything with respect to the ovarian hormone function. So there's a local hormone from the IUDs, a local exogenous ones, but there could still be ovulation. And likely still is ovulation from the ovaries, creating this very different hormonal milieu for IUD users and pill users, for instance. So we've talked about a lot of this before, and I know probably some of the other speakers are going to mention this. When I got into seeing, okay, what's going on with oral contraceptives and cognition, what does that literature look like? It's a bit messy. It's a lot messy. Like I said, I don't want to dwell on these different limitations. I just want to highlight one that continues to stand out to me with respect to the heterogeneity of those pills, right? I already mentioned they have a synthetic estrogen, but those progestins vary quite a bit. And even the days of active versus placebo pills toggle, the ways in which people use pills differ, right? Some folks don't use their placebo pills at all. And there was hints of this idea in the literature even 10 years ago, right? Belinda Pletzler and colleagues said that if OC users are heterogeneous with respect to, and they focused on the androgenicity of that progestin component, then basically saying we wouldn't know what to expect for behavior, right? If all these progestins have somewhat different actions and we combine them all, or combine individuals using them into a group and then say, okay, what does this group look like? We don't have a good sense of that. How could we possibly? And so we kind of took them up on their challenge in this first paper from 2015. And we looked at that mental rotations task in a variety of oral contraceptive users, right? This is the famous Vandenberg and Cuse task that we use a lot. We show participants that object on the left, and we say two of the objects on the right are rotations of that first object in three-dimensional space. Which are they? If you're trying to do it, it's the first and third in this case. And so when we took Pletzler and colleagues up on their challenge, if you want to consider it that way, we saw that in fact what they expected. If we compared, and this was a large sample for the time, naturally cycling women to men, there's a gender difference that we expected, with men outperforming naturally cycling women and men also outperforming oral contraceptive users. The effect size is a little smaller, but still significant. But if we then tried to consider those active ingredients, the difference in those oral contraceptive pills, and we grouped pill users according to, and we were in the US, so we used FDA criteria for what their active ingredients are. For us, these were some of the common pills that we saw that all had similar active or the same active ingredients, mainly ethinyl estradiol and the progestin norethindrone acetate. We also saw another group of users with triphasic pills, so they had varying levels of progestins, and in this case, the progestin norgestimate. And not in this first study, but in future studies and others have looked at these pills that have the progestin drospirenone. So these are all grouped according to their active ingredients, and these active ingredients, right, these progestins also seem to differ, or they also do differ in their androgenicity. Norethindrone acetate is moderate to high levels of androgenicity, norgestimate more mild, and drospirenone is actually an anti-androgenic progestin formulation. So if something about androgens matters for mental rotations, then maybe we'll see a difference in these groups where we didn't see a difference in the full combined OC group. And that's the case here. So this is the data from the same study, but now we've just broken up those OC users into a group that have norethindrone acetate in dark green and the group using pills with norgestimate there in the light green. And so what you'll see is all of the groups still differ from men, but users of the pill group that of pills that contain norethindrone acetate, they were performing better than naturally cycling women. right? And so we thought that maybe then there's some role for androgens or at least androgenicity of progestins, I should say, in oral contraceptives. And others have found something similar with more androgenic pills, folks using those having better performance, and folks using drospirenone, that anti-androgenic pill, having worse performance. But that might not be the whole story, right? Androgenicity of the progestin is this idea that people kind of hooked into early, but there's a lot of other things going on with hormonal contraceptives. And even in this same study, one thing that we considered and looked at was ethinyl estradiol dose. So the dose of the estrogen in the pill, we saw, again, these are small effects, right? These aren't whoppers that we're looking at here. They're part of the story, but across all of the pill users, those using pills with higher doses of ethinyl estradiol also seem to have slightly worse mental rotations performance. And in fact, in a paper that came out just last year, Betty Hampson's group also had a similar set of findings. Instead of looking at ethinyl estradiol dose, though, they looked at estrogenic potencies in the pill, and they only looked at users during their active pill phase and found this same inverse correlation. Right, so this is an interesting set of findings regarding mental rotations performance, and a role for estrogen or synthetic estrogens in these pills has also been implicated in verbal memory. So one of the first studies on OC use and cognition, or one of the earlier and more widely cited studies, showed that in fact oral contraceptive users, regardless of pill ingredients, seem to have better verbal memory than men and women at low hormone phases of their menstrual cycle. And this is also a small study, but consistent with other natural experiments, including menopause and menopausal hormone therapy, kind of calling out a role for estrogen and verbal memory. Moreover, and recently, there's some suggestion that maybe these effects are organizational. So maybe the way I stumbled into this field 10 years ago isn't so far off, because there are some studies showing that women who used hormonal contraceptives at some point in their life, when they're in their 40s, 50s, and 60s, seem to still have better both spatial and verbal skills than women who never used hormonal contraceptives. And so these are early studies, and we need to consider all of these nuances, but these are some of the conversations currently happening regarding group level effects of hormonal contraceptives and cognition. But more about how it's going, these findings, even though I kind of presented them in broad brushstrokes, if you will, there's a lot of nuance in there. Right? We haven't talked about a lot of the other variations in the pills or in the tasks. For instance, some of those studies on mental rotations talk about overall task performance. Some talk about timing on the task. Some look at variability. Some score the task differently. So there's some variation in when these findings pop up. There's also large individual differences in users that we really haven't talked a lot about yet. Things that we know have been important and that we've heard are important throughout this conference series, things like age, duration of pill use, and a variety of other features. So we have this sense now that maybe something about androgenic progestins, maybe something about low estradiol is important for cognition, but it probably plays out in variable ways when it comes to the day-to-day lives of users. Can we get closer to that now? That's what we're trying to do. Right? So one way we're trying to get closer to that is by considering different aspects of behavior that are also important for cognition. For instance, there's this long-running, what was called the sex role mediation hypothesis. It would probably be called the gender role mediation hypothesis if it were originated today. But the basic idea here is that different aspects of masculinity, how one perceives their own masculinity and personality, there seems to be a positive relationship between that aspect of gender and this good old reliable mental rotation score. And in a sample of naturally cycling women, we found this small expected positive relationship between masculinity and mental rotation score. But we didn't find that relationship for oral contraceptive users. And they're shown here in green. Basically, there was no relationship between masculinity and mental rotation score. And this could be an overall blunting of an effect, which we've again heard about throughout this conference and seen in other ways. Or it could be that there's something about the heterogeneity of these pills that's important for this feature. And so this is one way we're trying to map heterogeneity in pills today, right? 10 years later, or after those first grouping studies. And so this has been mentioned also a couple times throughout this series, but I'd like to point out the Managing Contraceptive Pill Patients book by Richard Dickey. And I think there's a more recent version than this that has some co-authors as well. This is a fabulous resource because it allows scholars to look up different pill formulations and code different aspects of the hormone potencies or activity levels according to bioassays of those. They could be pills, but it could be other forms. It could be like the IUDs and stuff as well in human and animal tissues. And it's a great set of tables, a great set of guides. You can look these things up. We did that in a most recent study, and I want to show you a little bit about how it plays out. There's different ways to do this, but the way that we did it was by looking at the estrogenic, androgenic, and progestational scores and coded them on a 1 to 4. We created this like Likert type scale based on the codes in the book, which were either low, which was 1, low to intermediate 2, intermediate to high, which was 3, or high, which was 4. And you can see here, I've plotted the estrogenic, androgenic, and progestational codes for several commonly used pills, at least commonly used in the US, right? So we get the sense here that low estrogen is highly androgenic, orthotricycline, really low progestational activity, and Yaz, because it's using this anti-androgenic progestin, really low androgenic activity. So we were able to get these codes for all of the pills and assign them or code individuals in our sample according to them. And here's the correlations among those different activity levels. And I really want to point out here, estrogenic activity is negatively related to androgenic activity, moderate correlation, and high correlation, high negative correlation with progestational activity, but no significant correlation between androgenic and progestational activity, at least in our sample of over 200 users. But when we used these codes to see if they moderated, if they qualified that initial relationship between masculinity and mental rotations performance, we actually found that estrogenic activity and progestational activity did. Androgenic activity didn't, right? But the effects are small. We would expect them to be small, right? So here's the relationship between masculinity and mental rotation score. And you see the dotted line for low estrogenic activity, but high masculinity. Those folks are performing in high mental rotation scores. But if you have low masculinity and high estrogenic activity, their performance levels were somewhat similar, right? So there's different ways to get to the same mental rotation score, to get to the same endpoint. And for progestational activity, the story is a bit flipped, such that high masculinity and high progestational activity is related to high mental rotation scores. So I also want to point out that I graphed the mental rotation score on the 0 to 40 axis, which is a set of possible scores, to try to contextualize what the real size of these effects are. They're statistically significant, but they're not huge. And again, call out estrogen and progestational activity, but not androgenic activity when thinking about mental rotations. So then the last point about how it's going, continued, if you will. Things have been tough in the last year, tough for women, tough for pregnant people. And in the US, after the Dobbs decision, there's questions about IUDs. Would they be banned post-Roe? And this is a story that came out in mid-July. Fortunately for me, 3 days later, some ideas I was working on were published, which was, is IUD use this like new frontier, I say, for behavioral neuroendocrinology, because IUDs differ in their hormonal milieu so much from OCs, it gives us a great chance to understand some of these gendered and cognitive components. And so really the only way to do this well is to think about then individuals. If things like masculinity, the type of pill, the type of hormonal contraceptive matter, we need to understand what's going on in the lives of individuals if we really want to understand cognition. So if we have a study with a whole set of variables and people and many measurement occasions, what we usually do and what I've presented so far is just looking at a bunch of people and a bunch of variables, we average across them and we make some inferences. But we could also just focus on maybe one person, same bunch of variables, but many, many, many measurement occasions to understand what's going on within individuals. What type of data could you use for this? You might think of like physiological data, like Fitbits and smartwatches. My group does a lot of daily surveys following folks, 75, 100 days in a row to figure out what their daily variation is like. You can do this with cognition. You'll see here, we've also created daily we use them daily, mental rotations tests that can be used for those 75 or 100 days, and even imaging data, right? They're time series data. You're getting a picture of the brain every 1, 2, 3 seconds, and you could map that by individual. And so we did this for an early study of IUD use and the neural bases of mental rotations. We have this slow event-related design where we really pit 2-dimensional and 3-dimensional rotation against each other. You'll see there's a 3-dimensional stimulus on the left, a 2-dimensional stimulus on the right. Participants see these in the scanner and they have to say, which ones show the same objects, just rotated versions, and which ones show different objects. This is a great task by my colleague, Jane Joseph. And so while IUD users did this task, we did functional imaging. And just to show you, this 3D versus 2D contrast is important, right? There's unique neural structures, architecture, activation for 3-dimensional versus 2-dimensional mental rotations. And there's suspicion that it's really this 3-dimensional component where the gender difference lies in mental rotations, if you will. So that's why we really wanted to focus on this contrast. We compared a default mode network, which is these blue ROIs, to a purported mental rotations network, which is these black ROIs. And you'll see a lot of activation in those 3D greater than 2D regions for that mental rotations network. That's on average, though. If you look at individual IUD users, if you map their individual functional networks during this performance, you'll see something that's much more varied. So what's toggling through these slides here are individual networks for each of the IUD users in our study, where the solid lines are time-locked associations, the dashed lines are associations from one TR to the next. They could be positive or negative in magnitude, and that line thickness shows how strong they are. And so you'll see here a great deal of variability, some consistency, but a great deal of variability. Speaks to the importance of getting at individual level effects, but these individual level effects can also provide insight into something important at the group level too. For instance, if we focus on just those ROIs in the mental rotation network and the default mode network, you'll see over here within the mental rotation network, there was greater connectivity while completing this task, as you would probably expect. And if you correlate those network activities with endogenous levels of estradiol, progesterone, testosterone, as well as task performance, you'll see some interesting patterns emerging. This is a small sample, so that's why I don't have significance flags on any of this. I just want to highlight some patterns. And one is here, right? If you recall, with those OC codes we did, estradiol was negatively correlated with progestational activity and androgenic activity. Here, estradiol is strongly correlated with testosterone and with progesterone, and it's positive. So we see a very different hormonal milieu in IUD users and OC users, and we see that task performance here is actually more strongly related to progesterone than to either estradiol or to testosterone. And of course, it's highly related to connectivity within that mental rotations network. So some final thoughts then to wrap this all up. Hormonal contraceptive use, it's a vital part of reproductive health for hundreds of millions of people. It's also a natural experiment that can help us understand aspects of neuroendocrinology that matter for gender differences and variability related to gender that's important for cognition. Takeaways from what I showed you here: mental rotations performance has been linked to androgenic progestins, also low estrogenic activity, things like masculinity as well, but the effects are small on average. Some might persist. They might be large for some individuals, or they might be negative, meaning in the opposite direction for other people. There's great heterogeneity. Another takeaway is that the hormonal milieus of OC and IUD users vary notably. This matters for cognition. We need to code our OCs in a way that give us a sense of that heterogeneity so we can understand variability in OC use and among IUD users well. And there's significant future potential in taking personalized approaches, mapping personalized network, following individuals daily to figure out how all of these complexities play out in the day-to-day lives of individuals. So thanks to funders, thanks to anyone who has ever talked to me about hormonal contraceptives. We're a small group. We inform each other's thinking so much. And if there's any time, I'm happy for questions or comments, or also happy to wait.
Thank you so much, Dr. Beltz. That was a fantastic talk. I'm sure that there is a lot of questions that our audience would like to ask. Maybe what we would suggest is that we keep the questions to the end of the session, and we go through all the talks first. So without any further ado, I'd like to introduce you to our next speaker, Dr. Birgit Derntel, who is a trained psychologist and a CBT psychotherapist, as well as a full professor for innovative neuroimaging at the Department for Psychiatry and Psychotherapy in Tübingen Center for Mental Health at the University of Tübingen in Germany. Her research focus is on social and affective psychoneuroendocrinology, with a particular focus on women's health. So Dr. Derntel, when you are ready, the Zoom floor is yours.
Thank you very much. Thanks, Jessie. Thank you, Nafisa, for introducing me here. But thanks, Jessie, and all the other organizers for setting up this wonderful conference. That is really great. It's so wonderful to meet. And as Adrian pointed out, it's so nice to see actually the faces and the people we read the papers about. And the wonderful thing about being a sandwich is not only that I'm a sandwich child in my family, but also being a sandwich here is Adrian actually explained most of the things that I had wanted to introduce, so I can be very quick on the theoretical things and can dig into the research that we are actually doing. So, I mean, contraception, of course, that's the topic of the series, so I don't have to teach you anything about why it is relevant. Just see the numbers and see what we are actually using. Adrian, you've already showed this, so that's great. What I want to point out is that we here in Germany, so the country that I'm speaking to you now from, we have looked into, or others have actually looked into, how the women report what contraceptive method are they using. And we see a reduction in oral contraceptive taking females. So from 2011 to 2018, there was a decrease in women taking oral contraceptives. And there's actually an increase in women using condoms. And for the coil or the IUD, there was no difference between those years. And we were just wondering, is there an increase in concerns? I mean, media coverage in terms of oral contraceptives has been big. So there is, of course, an ongoing discussion, and there is particularly a discussion on the psychological side effects of oral contraceptives. And so why can they actually do this? So why can oral contraceptives actually have an effect on the mood, on cognition, but also, and I will talk more about the emotions now, so why can they do this? And I mean, this is what Adrian also pointed out. You have the natural menstrual cycle, but once you introduce oral contraceptives to this hormonal system, then you have the synthetic hormones, the exogenous hormones, and they're very potent, and they kind of really suppress the endogenous ones. So your profile looks completely different once you are taking oral contraceptives, and I think this has to be taken into account. And these hormones, also the synthetic ones, they work on our brain. And the receptor sites in our brain are actually there where the social brain, of course, is happening. So here you can see from Claudia Barth, and this was also used by Esther Ambrase, the kind of pictures showing us where are the estrogen and the progesterone receptors located in the brain. So this is one for the dopaminergic system, but there's also one on the serotonergic pathway. You see that they are all kind of working together. The receptors are actually located in the regions that contribute to the pathways, but that are also essential for social aspects. And this was nicely reviewed by Montoya and Van den Bos, where they actually showed these are the regions that cross-sectional studies, for example, showed differences between naturally cycling women and women taking oral contraceptives. And that's the hippocampus, the amygdala, striatum, the ventromedial prefrontal cortex, and the ACC. So when you compare where are the receptor sites and where did they find actually differences in functional activation, you can easily see that these are regions that have dense receptors, hormonal receptors. So yes, there might be a way of how these hormones are actually influencing our mood, our cognition, our emotion. It may be via binding to these receptors. And that was one of the questions that Ann-Christine Kimmig, a former PhD candidate and now a postdoctoral researcher in my group, and I'm happy to have her. She was asking for her PhD thesis whether women taking OCs actually differ from naturally cycling women in terms of empathy. So are there differences? And if yes, where are they? So what she did is she developed a new task to measure empathy, the Tübinger empathy task, because we were not satisfied with what we had before. And I mean, this was also self-criticism that previous tasks that we developed for empathy are not really working or measuring or have several confounds. So with this version, we hoped to kind of overcome some of the limitations. And what we have here is that you have a condition where you are asked, the self condition, how do you feel? So imagine I give you the situation, I go on a concert of my favorite artist, and in this situation, I feel. So how do you feel if you go on the concert of your favorite artist from very negative to very positive? But then, as empathy is about self-other distinction, how are others actually feeling in a certain situation? And what does this do to you? We also have to have other conditions. And here we have the friend and the enemy. And now you can think about what would you feel if you imagine that your friend or the enemy goes on a concert of her favorite artist. In this situation, she probably feels, we could assume very positive, but how do you feel? So how do you feel knowing that your friend or enemy is now in this situation? And so we think that we can here really also differentiate between the cognitive empathy, so the perspective taking. So what is somebody feeling in a certain situation? We can infer that by knowing about the situation, but also our affective empathy, and this is the affective sharing component. So what are my emotional responses to knowing that a person is going through this or is having fun? My enemy? Not so sure. My friend? Yes, of course. So this is our Tübinger empathy task. And this is what actually Ann-Christine used for investigating empathy in oral contraceptive taking females and also naturally cycling. And she did a behavioral study before And then now I'm only showing you the data of the fMRI study, where she included data from 111 women, 46 females taking oral contraceptives, combined oral contraceptives. But as Adrian pointed out before, that was a heterogeneity in terms of the progestins that they were actually taking. We were just looking for monophasic combined pills, but we did not select only one group or only one progestin. So, of course, heterogeneity, definitely. Then 37 women during the early follicular phase of the natural cycle, so when estradiol and progesterone are very low. And then we also included 28 females during the periovulatory phase, because their estradiol is elevated. And that's what we wanted to investigate, because it was suggested that particularly during the periovulatory phase, that women are more outgoing and more searching for emotional, social interaction. And there was one study that actually pointed into this direction. So that's why we chose these two control groups, or these two naturally cycling groups. Now, in terms of behavior, we do see a significant difference between the women in the periovulatory phase and those taking oral contraceptives, with reduced cognitive and affective empathy ratings in the oral contraceptive taking females. And you see here the values for the self, the friend, and the enemy. And of course, the enemy, the affective ratings are strikingly different to when we feel for ourselves, whether we feel for our friend. So, I mean, this worked. And this is also what we wanted to show, of course. But here you can see that irrespective of the condition, the oral contraceptive females had lower emotional ratings for cognitive and affective empathy. In terms of brain activation, we also saw differences between the groups in terms of that there was a stronger functional activation in the periovulatory group and in the early follicular group in comparison to the oral contraceptive taking females. And here I showed that the dorsomedial prefrontal gyrus, for example, and the temporoparietal junction, two areas that have been shown previously to play a role and to be essential for empathy. So they also, here we also see significant differences in activation, and in the graphs, they are actually showing you that the bold signal again is reduced or lower in the oral contraceptive taking females compared to the periovulatory group. So it's not only that in behavior we see this reduced empathy ratings, we also see in the brain that there is a reduced activation in the OC taking females in both areas, the dorsomedial prefrontal gyrus as well as the TPJ. And then Ann-Christine was interested in whether there is also an association between self-reported empathy and brain activation. And yes, so the more empathic concern the women were actually reporting, the more dorsomedial prefrontal gyrus activation we could see. And then Adrian pointed it out, we don't have much information on the levels of the synthetic hormones, but for this study, we actually determined them. So we analyzed ethinyl estradiol and also the progestins. And what we observed is that the more ethinyl estradiol the women had, the less dorsomedial prefrontal gyrus activation, which actually fits to the pattern that we see that the OC women show reduced dorsomedial prefrontal gyrus activation. So it kind of fits to a nice pattern, at least this cross-sectional study, and we always have to be careful. There's no causality here. It's just correlation. It's just associations that we are seeing, that we see a reduced empathy in terms of behavior, also on the neural basis, and there seems to be an association with the synthetic hormones, with ethinyl estradiol in the oral contraceptive taking group. Now that is cross-sectional, and cross-sectional has lots of problems and limitations, of course, and Ann-Christine was not happy, and we set up a longitudinal study, and that was actually the data I showed you is the first part of the longitudinal study, and we also have repeated measures designed for the empathy for the second measurement time point, but we have not analyzed the data yet. She's currently on vacation, but next week once she's back, she will work on that again. And so what I can show you is now that we have collected all this data, and we have 4 groups now. We have one group that is in the early follicular phase in the first measurement time point and in the second measurement time point. We have constant OC taking women, so women that were taking OCs on the first time point and on the second time point. And then we were also interested in those that change. So we have a group of women that were measured for the first time point taking oral contraceptives, but for the repeated measures time point, they had stopped oral contraception intake. So we really measured what happens once they discontinue to take oral contraceptives. And we also had a starter group that was measured before they were actually starting oral contraception intake, and then while they are taking oral contraceptives. And this, you can see here, the time range between these 2 measurements was 3 to 8 months, 3 minimum, but 8, it was Corona time, so it was not that easy to actually have all the measurements and collect the data. Nevertheless, Ann-Christine has data of 84 women where she has 2 measurement time points. We analyzed the endogenous and the synthetic sex hormones, and she was particularly looking now into the resting state analysis and the resting state scan here. So I will now present you the changes over time with the T1 minus the T0. And what you see here are the changes in progestogens. So here is the endogenous progestin levels. And here you see that there's particularly the discontinua and the starters that show a change. And of course, this is also repeated in the synthetic groups, where those that stop taking oral contraceptives have a strong reduction in progestin levels, and those that start taking oral contraceptives have an increase in progestins in their system. While there's not much going on in terms of naturally cycling women, of course, because they don't have any synthetic hormones here. So we have this change in progestogens. And then Ann-Christine sent up a representational dissimilarity analysis. So we actually calculated the differences here, and then also put up, it's a lot of correlations, to be honest. And what you have here is in the upper row, and I'm just looking for my mouse, you see here in this upper row, those are the naturally cycling women. So there's not much of a change in terms of hormonal profiles for the progestins here. But here, we have the starters, and there you can see that the higher the values, the more changes here, and of course we have changes now in the starters. And she also looked into the resting state. We kind of parcellated the brain by the Shen Atlas, so now we have 267 parcels of the brain, and then she analyzed here the resting state connectivity. And again, for the first and for the second, she correlated it and looked for how similar or dissimilar are actually these values. And once she's then correlating the hormone changes with the brain changes, we can see that there are several regions in the brain that actually correspond or that are correlated with these changes in hormonal profiles. And that's the middle frontal gyrus. You can also see the hippocampus here. You can see the fusiform gyrus that is changed, and they all changed with the progestins. So we also ran the same analysis for ethinylestradiol, but there was no significant result. So this here is about the progestogens and the change in progestogens. This is interesting. We are just starting to actually analyze this longitudinal data set. We have 2 fMRI activity tasks as well. So there is lots of information that is still Ann-Christine has to work on. But I still thought it's very interesting to see what are the changes and how are the changes reflected in the brain, and which role do the synthetic hormones actually play here. And yes, they play a role. So there are changes that we can analyze. So to discuss this first part on empathy, and actually to discuss Ann-Christine's PhD thesis and parts of the PhD thesis, I think the Tübinger empathy task is a nice way of investigating empathy. It's always a discussion how we are going to measure these complex social phenomena. Can we really measure that in an fMRI setup? So yes, of course, there's always a discussion. And I think it's something we have to try because we also want to investigate these complex phenomena. And she has replicated the reduced behavioral affective empathy. We've done that in a behavioral study with the Winger empathy task, and now she's also showing it in an independent sample with the fMRI task. So it seems that there could be a reduction, but both studies were for now cross-sectional. So let's wait for the longitudinal data and let's see how actually changing your hormonal profile affects empathy. For the synthetic hormones, I really believe that that is something we should consider in future research. We should measure them, because otherwise we are discussing something where we don't have a grasp of what is actually happening. Just analyzing endogenous hormones is not enough in hormone contraceptive taking females. So I think that is something to consider. And the reversibility, it's the first fMRI study where we actually look at the discontinuation of oral contraceptive intake. And that's interesting. And I think we have to further look into that. So what does it do to the brain if women stop taking oral contraceptives? What are long-lasting effects? How reversible is all of this? This is just data from 26 women. So, I mean, small samples, but that could be hopefully stimulating for others to also investigate this. And then we can compare, we can pool, and we can really see what is happening. Now, that was the oral contraceptive part. And now, as Adrian also showed you, there is IUDs. And so I also want to talk about IUDs. And here, Adrian already introduced to you the levonorgestrel IUDs, or the hormonal coil, but there's also the copper coil, and I'm not going to talk very lengthy about the copper coil. It's more about the levonorgestrel coil that we are interested in. And when I say we, it's actually the work, the PhD thesis of Zoe Berger, a wonderful PhD candidate in my group, who is investigating the LNG IUDs. And I will show you now what her PhD study is about. But when Zoe actually started to work in that field, there was not much available in terms of LNG IUDs and how do they actually work on women. And so she started with a systematic review. And there was one study on stress reactivity, so one, and I'm going to show you the results of that study. There are several studies on mental health with very inconsistent data because the groups are very heterogeneous who are prescribed LNG IUDs and who are using LNG IUDs. So that was not very consistent. What was more consistent is that the quality of life actually increased because most of the studies that investigate this aspect have clinical samples. So heavy bleeding women, that's the group that they are that have the LNG IUD prescribed. So yes, the quality of life is increased in those samples. Sexual functioning, again, inconsistent data, but mostly yes, it improves. And then the brain. No study. When Zoe did the systematic review, no study was available. I mean, thank God that changed with Adrian now, reporting about the IUD, but still one study. So, I mean, it's really, there's lots of things we have to investigate in the future, and where we have to join our forces to actually investigate this together. Now, most of the studies, not yours, Adrian, but the others, most of them did not assess sex hormones. So we actually don't know about any systemic effects of the LNG IUDs. We believe that there might be some, because there are also psychological side effects to LNG IUD use. So whether this is all just because of the local functioning in the ovaries, or whether there are some hormones that travel even to the brain and then respond or work on the receptor sites, that is something we have to find out. So the stress study I'm going to show you now was done by Alec Navi-Jute and colleagues in the Netherlands. And it is the only study that looked into stress and IUD. And they had 4 groups. So they had the LNG IUD users, they had the copper IUD users, they had OC users, and naturally cycling women. And they performed the TRIA social stress test. So that is actually state of the art in terms of stress induction in the lab. And once they did that, you can actually see that the groups differed significantly in cortisol reactivity to the stress task. And the red line are the LNG IUD users. So you can see that they have a very elevated cortisol reactivity to the TSST, while the naturally cycling and the copper IUD, well, they show some response. The oral contraceptive taking women rather show a blunted cortisol reactivity. Now, they also looked into heart rate. And again, for heart rate, you can see the red line, the LNG IUD users, they have an elevated heart rate response to the stress induction, significantly higher compared to the other three groups that did not differ significantly from each other. So what this study is actually proposing, and they also had hair cortisol, and they had another study where they investigated the HPA axis. What this is actually proposing is that LNG IUD users, and it was also pointed out by Adrian before, but I think you can all imagine now, you cannot say hormonal contraception is working in this way, and now we can combine them, but at least for stress, we cannot combine IUD and OC. And of course, it's interesting, is one study, can we replicate this? And in this study, we didn't have any information on the sex hormone levels, so there were no sex hormones assessed. There was also no information on intake duration and no subjective stress information. So we don't know how stressed actually the females felt while undergoing the stress induction. And that's now all part of Zoe's PhD study. Up to now, she has collected data of women using LNG IUDs, of women using OCs, that's again monophasic combined OCs. Again, we are not just going for a levonorgestrel, we are actually combining, so it's a heterogeneous group. And we have the naturally cycling women measured during the mid-luteal phase. They are actually performing, so that's the study design, and they are performing either the MAST as a stress induction or the placebo MAST, and we have relaxation periods. You can also see that we are taking saliva samples, of course, to estimate the cortisol reaction. We ask them for their subjective ratings, and we take a blood sample to analyze the sex hormones then. Now, the Maastricht acute stress task is a very nice task that we like to use in the lab. What happens here is that you ask actually the women to put the hand in the bucket with ice water, and you also have them count backwards. So there is a physiological stress, there is a achievement stress, and there's a social evaluation because you also have an experiment and a lab code. And for the placebo condition, that is all, of course, a little bit different. Now it's not ice cold water, it's lukewarm water, and they don't have to count backwards, they just have to count to 25. That's the sample description. And I don't want you to look at all what we are assessing, it's just groups didn't differ in many aspects, also in age and other interesting variables. But what they actually show is that in terms of subjective feelings of stress, the MAST is successful. We do see that they feel stressed after performing the MAST and not the placebo. And a similar effect for anxiety, so they feel more anxious after doing the MAST than the placebo. We also look into positive and negative effect. For positive effect, no group difference. For negative effect, there is a group difference and there's a task by time difference. So the MAST again is increasing the negative effect. And we also see that this is particularly high in the IUD users. And then cortisol reactivity. And please, it's only preliminary data. We are waiting for the final data analysis. So what I show you now is really, really preliminary. And it's only 15 IUD, 6 OC, 5 NC. But what we see up to now is that it's particularly after the stress induction, the IUDs respond with the cortisol versus the two other groups. Not so sure, but we have to wait for the final analysis to actually say more about what's happening here. So in summary, what the IUDs are actually doing, the subjective stress is significantly increased. It's worse after the Maastricht acute stress test and not so bad after the placebo. So it's a successful stress induction. Our IUD users have the significantly higher negative effect. And also from the data that we have here, It's also that the cortisol reactivity is different. It's higher. It's actually what we would have expected based on the data from Alec Navi-Jute. So this seems to be in line with that first study on stress reactivity. Now, what are the implications? I think this is for both, actually. It's the more we know about the psychological side effects of hormonal contraception, the better we can actually inform and the better we can actually deal with the concerns of the women to kind of what is your contraceptive choice? What would be the ideal contraception for you? But it will also help us in actually developing new contraceptive methods, probably, that then we can also advise. Because ultimately, our goal should be that all women should be able to reliably control their fertility, but not having to potentially compromise their mental health. So this is something that we are, of course, investigating in Tübingen, and not only in Tübingen, we are also doing this with our colleagues from Uppsala University, and we've just been funded an international research training group on women's mental health. And here you can see the PIs, and one of the Mercator fellows I've also highlighted, because Lisa Galea, you've definitely met throughout this series, and she's also advising us on this project. And I want to thank the collaborators, the funders, my group, in particular, Ann-Christine Kimmich and Zoe Bürger, for doing this wonderful research, for working that many hours to kind of get the data that we need to inform other women about their contraception. And my last slide, we are recruiting. So we have one project in the IRTG where we are looking for a PhD candidate, and it's a project on maternal stress and fetal imaging. So we're measuring the fetus. And we also have another project for a PhD candidate where we are applying transcutaneous vagus nerve stimulation in the postpartum period. So if you are interested, or if you know anybody who might be interested, please forward this information. We'll be happy to receive applications. Thank you.
Thank you so much, Dr. Derntel. That was a fantastic talk. Thank you for sharing your amazing data with us. So I am happy to introduce you to our final speaker, Dr. Marie-France Marin. Dr. Marin holds a PhD in neuroscience from the University of Montreal and completed her postdoctoral training at the Massachusetts General Hospital at the Harvard Medical School. She is a professor in the Department of Psychology at the University of Quebec in Montreal, an associate professor in the Department of Psychiatry and Addiction at the Université de Montréal, a researcher at the Montreal Mental Health University Institute Research Center, and a research member of the Réseau intersectionnel de recherche en santé de l'Université du Québec. Her expertise focuses on the impact of stress and stress hormones on the modulation of emotional memories. So without further ado, Dr. Marin, the Zoom floor is yours.
All right, thank you so much. Can you see my slides?
Yes, we can. It's perfect.
Great, thank you so much. So I want to thank you for, I want to thank Jessie and others for organizing the symposium. It's great. It's great also to see people that we read in the literature and put faces on these papers. And I'm really happy to be here today to share some results from the lab. It's very fresh results. Most of them have not been published yet. But I first want to start by giving a lot of credit to two of my wonderful graduate students, Alexandra Bouillard and Lisa Marie Davignon. They've been working really hard on the data that I'll show you today. And just to give you a little bit of background, our lab is mostly interested in understanding the biological and environmental influences that modulate fear expression and fear regulation. So we are a fear lab, and we also like to stress people. So we make people fearful and stressed. And we are interested to see how this modulation and how we perceive fear and how we are able to regulate it can contribute to one's vulnerability to later develop fear-related psychopathology, such as anxiety disorders and PTSD. So just to begin a little bit, to give you a bit of background, why are we interested in studying fear? So fear is a universal emotion. It's very important in guiding our behaviors, either by approaching or by avoiding certain situations. And we know that both fear expression and fear regulations are regulated by the interplay of various brain regions, such as the amygdala, the hippocampus, the dorsal anterior cingulate cortex, the insular cortex, and the ventromedial prefrontal cortex. Of course, it's very complex and all those regions work together, but these regions that I just mentioned have been identified as key hubs in the brain as modulator of fear expression and regulation. And we can study fear in the laboratory using various protocols. We favor the fear conditioning and extinction protocol because it allows us to study how people learn to associate fear with a neutral stimuli, and then to see how they are able to later regulate their fear. So these are the regions involved in the fear circuitry that I just mentioned. We're missing here the insular cortex, but as I mentioned, these regions are, yes, important for fear. They are filled with sex hormones receptors, and that's why they've been studied a lot in context of fear learning and expression. But these regions are also important for various cognitive functions, and we'll get back to that later. So just a little word on how sex hormones, affect fear processes. So this studies has been conducted by Graham, the reference is wrong actually, is Bronwyn Graham and Mohamed Milad. And what they've shown is that when we study women that are varying based on estradiol profile. So when we study women with a low level of estradiol, high level of estradiol, or a woman taking hormonal contraceptive, what we see is that women, irrespective of their status, are able to acquire fear. So during the fear conditioning process, there are no differences between these two groups of women. At the end of extinction, so what we do during extinction is that we present the cue over and over, and eventually people will learn to regulate their fear level. What we observe is that, again, no differences between those two groups, suggesting that women can learn efficiently to regulate their fear, at least within a specific session. What we are mostly interested in is the transfer of this safety memory that is being formed during extinction learning. So during extinction, we learned that a stimulus does not predict a aversive stimulus anymore. So for example, a light does not predict a shock anymore. And what we want to be able to see is whether we give people a short break and they come back, for example, 1 day later, are they able to recall this safety memory, or will the fear memory be expressed once again? So when we bring back people in the lab 1 day later, what studies have shown, this one and others, what they have shown is that women who have low level of estradiol, so naturally cycling women early in their cycle, or women using hormonal contraceptive, so who also have low endogenous levels of sex hormones, both show higher fear recovery, so more fear expression, when tested during extinction recall compared to women with high estrogen level. What's interesting here is that men perform similar to women with elevated levels of estradiol, suggesting that it's really the low endogenous levels of estradiol in women that is problematic here. So this is at a psychophysiological level, so using here skin conductance responses as an index of fear. Obviously, there are other ways to study fear. We can, it was mentioned in a previous talk that we can ask about subjective reports, what people, how fearful are they of a given stimulus, but we can also study fear using brain structures, of course, and that's what we're doing in the lab. Before moving to our data, I just want to give you a little bit more feedback, but my colleagues have done a wonderful job before, so I'll go quickly. We know, as I mentioned earlier, that the brain regions that I've showed you are involved in various cognitive functions. And a lot of these functions have shown important sex differences. So when we compare men to women, and of course, there can be some gender influences and there can be some biological influences. Shortly, what the literature has shown over and over is that men tend to perform better on tasks involving spatial perception or mental rotation. And this effect seems also to be modulated by testosterone, such that higher level of testosterone are linked with better performance. We also know that women have a tendency to perform better compared to men on tasks that involve verbal skills. So whether it's verbal fluency, verbal memory, and this effect also seems to be, at least to some degree, biologically driven, because estradiol levels have been positively associated with these performances. And what we know also is that oral contraceptives modulate this effect. So verbal fluency appears to be negatively affected in women using contraceptive compared to naturally cycling women. So basically, women who are not using the pill show better verbal fluency skills that are higher than men, and COC user will be falling in the middle. So that's what the literature has shown us. Obviously, there is some contradiction in some of the results. That's science. And so knowing all of this, we were interested in examining both the current but also the past effect of using the pill. So whether you are using the pill now, whether you have used the pill in the past, we were interested in seeing how this affects fear circuitry. As I mentioned, it's impacting fear a lot, both in terms of expression, but also in terms of neural correlates. We were also interested in cortisol secretion. I will not be talking about cortisol today, but just so that you know, we are following cortisol here, but we are looking at the diurnal cycle compared to the talk just before that showed data using psychosocial stressor. And finally, we were also interested in investigating these effects on cognitive functions. So we began a study right when the pandemic happened. That was wonderful. And what Alexandra and Lisa Marie did is that they recruited a sample of more than 200 people. And this sample was composed of the following groups. So we had men, we had naturally cycling women, and we had women actually taking oral contraceptive, and we have women who had used oral contraceptive in the past and had stopped using it. All the women that were recruiting in the study were, well, all the women who have used or were currently user, it was all combined oral contraceptive. So a mix of synthetic estradiol with progestin. So we didn't have IUD, we didn't have the progestin only pill. So it was all combined for this study. And we collected a lot of data, of course, on variables such as sociodemographic variables that could distinguish our groups. So for example, age, education, religious beliefs, we collected information about being in a relationship or not. So all these variables, but we also collected information in current and past users about specifics of their consumption, basically. So we look at the age at which they started using a COC. We look at the age at which they stopped using COC for the past users. We also look at the duration of use. We check whether they took breaks. We look at the different generation of pills that they have been using. So we have all these information. And of course, today I'm showing some of the results. As I said, these are not published. We are at the beginning of getting our hands on the data right now. The data collection is over, but this is just to give you a hint, and we can discuss later during the question period, of course. But the idea here is just to show you a bit what we are interested in. All these women and also a group of men came to the laboratory for 3 different visits. So at the first visit, they fill out various questionnaires assessing, for example, psychological traits. We were also interested in gender, so various questionnaires, and they also perform cognitive tasks that I'll describe in a moment, but basically the tasks that I mentioned earlier that have shown a strong difference between men and men, so mental rotation, verbal fluency, and verbal memory. They also took saliva samples at home in order to quantify diurnal profiles of cortisol, and they fill out different questionnaires at home regarding the collect of cortisol. And they came back for the second and the third visit at a different facility where there was a MRI scan. And during these 2 visits, so the second and the third visit, we basically put them in the scanner, and we did a fear conditioning and fear extinction protocol. And we also acquired structural data in the scanner as well. So while they were in the scan performing the fear conditioning and extinction protocol, we also track skin conductance responses. So basically, this is the study. As you notice, we have naturally cycling women. So either they are never users of COC or past users, but are now naturally cycling. So for these women who had a cycle, we asked them to come, about half of them came in the early follicular phase, so when estradiol and progesterone were very low, and half of them came when in mid-cycle, so we wanted to mostly modulate estradiol levels, so they came when estradiol was elevated, but progesterone was expected to be low at mid-cycle, and we did the reverse. So if you come for first visit in the early follicular, you come for your second and third visit in the in the mid-cycle phase, sorry. The second and the third visit were separated by only 1 day, whereas the first and the second was separated in average by about 2 weeks, but it depends, of course, on the participants. So this is our design. Now, let me show you some of the results with regards to the first visit. So the cognitive tasks that were used. This is Lisa Marie. I mentioned her earlier. She's doing her PhD right now at UQAM in psychology, and she's interested in that portion of the grant, I would say, of the bigger project on cognition. She will also use the physiological data and fMRI data for the fear protocol, but we haven't looked at those data yet. So today I will talk to you about cognitive functions. So Lisa wanted to document the sex differences, but also the role of COCUs on mental rotation, verbal fluency, and verbal memory. So let's start with mental rotation, sorry. So in terms of mental rotation, we use the MRT task, and here we show a strong, we have a group difference where men show the best performance, OC users are in the middle, and NC have the lowest performance. And the significant difference here, as you see, is really between the group of men and naturally cycling women. What I have to tell you, you see the sample size for each group below. What I have to tell you is that the naturally cycling women here, we combined both groups. So we combined those who came during the early follicular and those who came during the mid-cycle. But before combining, we made sure also that they were not showing any difference in terms of performance. We were surprised, but that was not the case. So we were able to combine them. We also have saliva samples that were taken at each visit at the scan and for the cognition. So we will be able to verify in terms of sex hormones whether they do modulate the effect. So we quantify endogenous estradiol levels, we quantify progesterone levels, although we expect them to be low. We quantify testosterone, and we also quantify phenylestradiol in those samples. So here, this aligns well with what has been shown before in the literature, where men show the strongest performance compared to naturally cycling women. And unfortunately, although the OC group is not showing any difference between men or NC, we kind of see that they are in the middle. In terms of verbal fluency, we use the three different scales, the phonemic, semantic, and switching, and we combine them in terms of number of words that they generated, because there was no interaction with the type of block that we use. And there was no group difference. However, when we look within the group of OC users, what we see is a correlation between the number of years that people have been using the pill and the number of words that they were able to generate. So basically, higher performance is correlated with a longer duration of use. We also look at it in a different manner. So when looking at the scatter plot, seems to be 9 is about the median. So we did a group of people, of women who use COC for 9 years and less, or those who have been using it for more than 9 years. And what we wanted to see here is to compare them to the performance of naturally cycling women. So the idea here is to see whether those who have been using it for a few years, are they impaired compared to naturally cycling women, or it's really a function that improves over time that seems to be almost linear. And so by putting the naturally cycling woman in the equation, what we see here is a clear improvement with COCUs, that where the advantage is really starting to show around 9 years of use for the COC. Finally, we also look at verbal memory. For the verbal memory, we use the re-auditory verbal learning test. So as you know, there are 5 trials that are very close in time, testing basically the learning curve. So there was no difference between groups in terms of learning, but then there are 2 trials that have a little delay, so that allow us to test delayed memory. And what we see is an effect of group emerging only at the trial 7. So when there's a delay, OC the OC group show here an advantage in terms of verbal memory over men. So this results converge with the previous one, basically showing that OC use has some advantage in terms of verbal skills, both memory and fluency. So these are the results that we have for cognitions. We are also investigating so that you know whether there are differences in terms of androgenicity, so the generation of COCUs that is being taken by the woman. We are also investigating the effect of both endogenous and exogenous sex hormones. I will now turn to the second day. So on the second day, as I mentioned, women and men are coming to the lab. They're going to the MRI facility in order to acquire both structural and functional data. And Alexandra Brouillard, who's here, is also doing her PhD in psychology at UQAM. And Alexandra is really interested in the structural MRI correlates associated with COCUs. So she wants to examine the role of endogenous and exogenous sex hormones on structural correlates of fear circuitry. And her thesis is really about structural MRI. So I'll show you a part of her thesis and I'll give you a hint on what she's analyzing also for the next phase. So as I mentioned earlier, we are documenting, we are actually collecting saliva samples at the lab at session 1 and session 2. And so here are the data for the second session, so when they are at the scan. So these are, so for A, you have the estradiol levels, you have progesterone in the middle and testosterone on the right. And the idea here was to see whether we can separate easily, to identify easily our group based on hormonal concentration, and not only rely on the self-report of women, of course, and men. Well, men, there's no cycle, obviously, but so for estradiol, as you can see, men and COC users are very low level. All women, so the gray, the dark gray are women who are past users, and the light or almost white bars are women who are never users. And these were about half and half different based on the phase during which they came. So those who came during early follicular have very low level of estradiol, which is expected, but they don't differ based on their previous status. And those who came during mid-cycle had higher level of estradiol, again, that was expected, but our groups based on the status of never user or past users don't differ. In terms of progesterone, sorry, all groups do not show any difference, and that was expected. We were recruiting people at time where progesterone is supposed to be very low, so this pattern was expected. And finally, men showed way higher testosterone level than our groups of women. So that confirms basically that the recruitment based on self-report in terms of menstrual cycle phase work well in this case. Now, Alexandra is interested in the structural data, so she's looking at the fear circuitry that I showed you earlier. Of course, when doing fMRI and MRI data, there's always a big debate as to how we should analyze our data. Should we go with a region of interest based approach? So we just go with Roy's and we look at where we have a priori hypotheses, or should we go all brain? In this case, we will investigate both, but I will focus today on the regions of the fear circuitry, because we know that they are rich in sex hormone receptors. So that's why we had a priori hypothesis on these regions. So she's interested in both volumes, but also for region, for cortex region, she will look at cortical thickness. In terms of volume, so I just want to remind you what are our regions of interest. We look at amygdala, hippocampus, ventromedial prefrontal cortex, dorsal anterior cingulate cortex, and we also look at insular cortex. So here you see the dorsal anterior cingulate cortex. We found a main effect of group in these regions for the volume, irrespective of the hemisphere. So it was the effect was similar on both hemispheres. And what we show basically is a strong sex difference. So all group of women, irrespective of their history of COC use, differ from men, so add bigger volume of the dorsal anterior cingulate cortex. I just want to remind you that this region is particularly important for promoting fear. So when we do, for example, a fear conditioning task, the DACC will activate, and usually the higher the activation of the DACC, the more fear expression. And it's also been shown that the thicker this region, the higher the fear expression as well. So that's results for volume. Now, in terms of thickness, there was a group effect in the ventromedial prefrontal cortex. And this region is very much important for regulating fear. What we show here is a difference between men and women who are currently using the pill. So as you can see, never users and past users fall right in the middle, and they don't differentiate from the two other groups. What we see here that current user had a thinner ventromedial prefrontal cortex, thinner cortex of the ventromedial prefrontal. So that's really interesting because this region, as I mentioned, has been involved in fear regulation. And so what it suggests is that studies have shown that the thinner it is, the more you have difficulty regulating your fear. So here we show a sex difference in terms of a region of fear expression, but we also show that the women who are currently using the pill have a thinner region in where it's very important to regulate fear, which can suggest some sort of vulnerability in terms of fear regulation in current OC users. Alexandra is right now investigating also the I would say the parameters associated with COC use. So she's looking at how the age at which women began using a COC and the duration of use can modulate these structural correlates of fear. So as I mentioned earlier, when we began the study, the COVID happened. So we had to do something. We had very few data at the time, but we had data outside the scan from different studies. And we have always been interested in sex hormones and stress hormones. So we have been documenting the history of contraceptive use and all of that in our subject. And of course, as you know, the COVID happened and it had a profound impact on mental health for a lot of people, and a lot of data have been published showing that women displayed more symptoms of distress compared to men. So we were interested here in investigating whether hormonal contraceptive use could modulate this vulnerability factor of distress among our women. So what we did is that we called back participants from our lab who previously participated in different studies. And we asked them whether they were interested in taking part in a longitudinal investigation. So they took part in the first study that happened before the COVID. COVID happened in mid-March. We had our first confinement in Quebec, and we obtained IRB approval. Took a little bit of time, and then in June we start to send them different links. Basically, they did it online, where they fill out information about distress. So we assess depression, stress, anxiety symptoms, and PTSD symptoms in these individuals. When they were recruited before pandemic, they were all healthy. So we were not expecting here psychopathology level, but we were expecting, of course, variability in terms of distress, because we were all at some point during this pandemic under a lot of stress. And so some symptoms appear without necessarily reaching the pathological state. And so every 3 months, we send them the same questionnaires, basically. So we ended up with 49 men and 32 women who were using whole contraceptive. Well, here was hormonal contraceptive. So we combined all types because we were not able to, we were not sufficiently powered to make the distinction, but basically it was about 2/3 who were using combined oral contraceptives. And we also had 73 women who were not using hormonal contraceptives, so who were naturally cycling women. And as I mentioned, we assessed those symptoms. We scanned them. I'm not expecting you to read this slide, and I see the time passing, but we scanned them on various sociodemographic to make sure to control for potential group difference. So for example, age, education, personality traits, stressful events, and all of that. So we controlled for everything that show a trend of between group difference to make sure that we cannot explain our results by these potential confounding variable. Here, what I'm showing you basically is that in general, as you see, this is the DAS total. So all symptoms of stress, anxiety, and depression combined. And here it's by subscale. So you have the stress subscale, the anxiety, and the depression. When you have the bar graph, it's because there was no interaction with time. So overall, over the year, the effect was the same. And mostly what we see is an effect of a sex difference that is mostly driven by the hormonal contraceptive users, where they show higher anxiety, higher stress level than NC user and then men. And there was no difference in terms of depressive symptoms. But what's very interesting is that among our naturally cycling women, I said that we had 73 naturally cycling women. We had past users and we have never users. And so based on that, we did a preliminary, or I would say exploratory analysis, because we don't have a lot of women, but basically we split that group. So among our women, are we able to see long-lasting effects of the pill? And the answer is yes. So again, these are the same, the total scale, the anxiety, the stress scale, and the depression. No effect again in depression. But what we see is that the current users have higher level of anxiety, and these levels are similar to our past users. Both of these groups show higher level of anxiety compared to the never users. And we see a similar pattern for stress, although the significant difference is only between the current users and the never users in this case. So overall, what I've showed you today in terms of COC use and cognition, that it's important to not only consider COC use per se, because yes, we can find group differences, but it seems to be important also to consider the duration of use, because some effects might appear as a function of that. So more and more we need to consider, of course, the parameters associated with COC use. In terms of structural data, I've showed you, yes, an effect of sex in the DACC, but also remember that the current users of COC have thinner ventromedial prefrontal cortex, which is a region important to regulate emotion, especially fear. And finally, I showed you that preliminary results that suggest maybe a long-lasting effect of hormonal contraceptives in terms of distress. This was in a longitudinal study that lasted a year. So I just want to thank the different organisms that are funding our work. And again, special thanks to Alexandra and Lisa Marie for doing all this work.
Thank you so much, Dr. Marin, for this fantastic talk. I would like to open the floor to questions now. We have a few minutes. I have a question for you, Dr. Marin. I loved that you compared women that had discontinued using oral contraceptives and those that never took it. I think that was a very important comparison, and it was striking to see that there were long-lasting effects of usage. And I'm wondering, do you know, like, what was the range of discontinuation of usage?
In terms of how long have they not been using it?
Yes, yes.
We didn't have a lot of information on that because it was all retrospective. In the current study, though, that we are conducting with the fMRI and everything court and cognition, there we ask basically all questions that you can think of, but we are following now for how long. The thing, though, is that in the study that we are currently conducting, we only included women who have been who have stopped using it for more than a year or 18 months. I can't remember. So we might miss, you know, the potential lasting effects. We wanted to be free of COC, but maybe we shoot ourselves in the foot. It's unclear. So, but we made like a clear distinction with past and current. Yeah.
Okay. Okay. Thank you so much.
Pleasure.
We have a question from Karina. Hi, Karina.
Hello, everyone. I have a question regarding the talk on the IUD and the enhanced cortisol levels. So there are two questions. The first is, are there any dose-dependent effects? Like, for example, people using the Mirena or the Jadis, which have a great difference in dosage regarding the levonorgestrel? That would be the first. I can also continue with the second, and then you can answer at one. Okay, perfect. And the second question would be regarding the enhanced cortisol levels. I was wondering if there might also be an effect from progesterone being like a preform of cortisol. So the cortisol synthesis can also be like biased by enhanced progesterone levels. That's what I thought, that maybe that this can also lead to enhanced cortisol levels because it's a preform of cortisol, but I don't have any data on the synthetic prostogens, how they affect that.
So thank you very much for the questions. Actually, in terms of dosage, I mean, you saw it was only 15 IUD women. Of course, we note down. So in the end, we'll have more. But for this cortisol, it was only 15. So I'd be very careful to actually say that that is the final result. And second, yes, we noted down which IUD the women are using. So in the end, we can actually see whether there's a difference of the Jadis or the Mirena. So that is what we will have in the end. But for now, we didn't look into this. It's just 15. So we're waiting for the final data. Actually, so we received them today. So let's see what happens once we analyze that. And in terms of what about progestogens or what about the IUDs and how do they work on the cortisol? I mean, that is a question still under debate, whether the IUDs have a systemic effect, yes or no. There are several that say, no, that's not the fact. They're just working in the ovaries. But there are more and more studies actually showing that there are systemic effects. And I think also because of the side effects that we definitely have to talk about these effects and definitely have to investigate them any further. How levonorgestrel is actually influencing the cortisol and the HPA axis, because it's not only cortisol, there may actually be precursors that are more influenced by that. I can't tell you actually. I'm sure that there are some studies available, and Zoe is also online, hopefully. So I mean, she could probably jump in and tell you more about this. But I think we're just starting to understand what's happening here, because we're starting to actually investigate the women with IUDs, also in terms of neural activation, as Adrian did, but also in terms of psychophysiology and what is happening here.
Thank you very much. Looking forward to the results. Thanks.
Me too. Thanks.
Thank you. There is a question in the chat that I'm going to read. Regarding discontinuation, Dr. Derntel, I like the approach you described to identify the possibility of reversibility. On this point, a question for Dr. Belt. Have you done any studies looking at the discontinuation of the pill and the 3D mental rotation task?
Yeah, thanks for that question. I think we're in the no, we haven't done any yet. We are in the process of collecting data on the topic. So stay tuned. Although given data we've seen today and some of the data I presented in sort of the organizational framework of how these hormones might act, I would have the hypothesis that some effects would persist. I think the interesting question about discontinuation or reversibility, just like we talk about duration of pill use, we have to talk about the gap then between, and I think this was referred to earlier as well, we have to talk about the gap of the time, like a potential washout period. You know, is such a thing possible over long periods of time? What is that time? Or are some of the effects potentially organizational and maybe they don't weaken much with time? And I think the answer to these questions right, is likely going to be with, as it is with many of these aspects of cognition, it depends. It's going to depend on the person, it's going to depend on the cognitive domain, and a variety of other factors. But I think it's such an exciting space to be in right now, because so much of all of our work, you would think we prepared our slides in like an overlapping way, but we didn't. We're all like working along similar lines, coming to similar conclusions independently. And I think that's such a great sign for our science. So these questions about reversibility or organization, however you want to frame them, they're one of the next steps for us in human work. And hopefully animal studies can help guide some of these questions, particularly in a causal, more mechanistic way than we can do in humans.
Thank you. Can I ask a follow-up question? And this could be for any of you. Is there a particular approach that you would use to look at individual variation compared to the average variation? What kinds of approaches would you use?
Yeah, I'd use an intensive longitudinal study. If you want to get at individual variation in cognition, it's tricky, right? It's a tricky business because you need to be able to use cognitive tests that have parallel forms established, so that you don't have practice effects with a given form. But even then, in most cognitive domains, you'll see a little practice effect, but it depends on domain. I can tell you some things in my group. We have intensive longitudinal measures for mental rotations, for verbal recall, and recently for inhibition in like a Stroop-like task. And you see different levels of learning or practice effects initially, usually about a week to 10 days. And then you start to see individual level fluctuations to different degrees, depending on the person and the context. But some of the things we're interested in then, and I think this alludes maybe to the next question in the chat, it's not only hormones that matter for cognition. Hormones matter for cognition, for some people at some times in different ways, but daily experiences, maybe lifelong experiences, you only capture cognition. I'm a developmentalist by training, and you think if you only capture cognition in a moment, but there's a lifetime of experiences that end up reflected in that moment, plus that context. So capturing those individual variations, how they differ from the average, means you need a lot of individual assessments. How many, depends on the question and this sort of thing, but you need many individual assessments, and they have to be good assessments so you can separate learning from these other factors.
Thank you so much.
I could talk about this for a long time.
Thank you, Dr. Beltz, and thank you, Thomas, for your question. Maddie, your question is amazing in the chat, and I'm sorry that we didn't get to formally answer it, but I'm glad that at least Dr. Beltz was able to touch upon it. And so don't hesitate, if any of you have questions, to write directly to our speakers. I'm sure that they'll be very happy to answer your question. And with that, I would like to thank everyone for attending, for your great questions. We truly hope that you enjoyed this event. Our next conference event is on the slide. You're able to register for it if you are interested in attending, and you'll be receiving a link for it. And we look forward to seeing you at this event. Thank you all for attending. Have a wonderful day.
Thank you so much to the speakers. It was an excellent symposium today.